After learning to make a thin blood smear, the smear must be stained in order to see the cells. Smears can be stained in a number of ways. Many laboratories have automated stainers. There are a variety of different types available. In the student lab, you will stain your smears using a manual method by dipping your slides in the staining solutions. The Wright stains is the most commonly used stain in the hematology lab and is composed of two different stains and a buffer because the staining process is pH dependent. The two stains are eosin and methylene blue. The eosin attaches to negatively charged ions in the cell and is red in color. The methylene blue attaches to the positively charged ions and is, of course, blue in color. The buffer maintains the pH of the solution to ensure proper staining. The slide is then dipped in methanol to permanently adhere the smear to the glass slide. This process is known as fixing the smear. After dipping the slide, it is allowed to air dry. The next step is to dip the smear 5 times in the eosin solution. In Copeland jars, there are ridges to keep slides separated from each other. For our purposes, you will not be using them, so dip at an angle and run the edge of the slide along the edge of the jar while removing it from the stain. This allows excess stain to be removed from the slide. Then, dip the slide 5 times in methylene blue. The slide is then rinsed in deionized water and is agitated to remove any excess stain. You want to make sure that you remove any stain that has not bound to the cells, but if you rinse for too long, you may leach out the stains from within the cells. Generally, rinsing the slide for 6 to 10 seconds is adequate. Allow the slides to air dry before examining them under the microscope or applying a cover glass. When using a quick stain, the pH cannot be controlled, but the staining can be adjusted by changing the slide's exposure to the stain by modifying the number of times the slide is dipped into the stain. When examining the stained slide, if the cells are too pink, you have too much eosin being bound, so the stain is too acidic. Adjust by dipping the slide fewer times in the eosin. If the slide is too blue, the pH is too alkaline, and you can adjust by fewer dips in the methylene blue.